The cell's life is divided into distinct stages within the cell cycle. The cell cycle are events that occur during a cell's life. Basically, the cell grows, more organelles, more cytosol, and expresses vital genes to create proteins that control the stages of cell cycle. It copies DNA and it grows even larger with more plasma membrane and more vital proteins. Additionally, mitochondria divide to ensure the production of ATP to power the activities required for successful cell division. Eventually, the cell needs to divide into smaller cells, but to do so, the chromosomes need to divide within the nucleus. Once two nuclei are formed, the cell membranes can pitch in and two new cells are formed. There are both internal and external control systems that ensure that cells proceed through the cell cycle at the correct rate, and less damage is done to the genes that code for protein that govern the cell cycle. It's divided into interphase and M phase, mitosis and cytokinesis. Interphase is divided into G1, S, and G2, and accounts for 90% of a cell's life. M phase stands for either mitosis or meiosis and means nuclear division, then cytoplasmic division. Cytokinesis means division of the cell itself. In G1, the cell grows proteins such as cyclin, which causes cell growth. When it reaches the G1 checkpoint, it's checked for nutrients, growth factors, and DNA damage. After G1, if the cell is not allowed to enter mitosis, it enters G0, which is the resting state. In S phase, DNA is duplicated. S phase stands for synthesis, and there's no S phase checkpoint. In G2, CDK binds to cyclin, which produces MPF, which then phosphorylates mitosis, which is the next phase of the cell cycle. The G2 checkpoint checks for cell size and DNA replication. The next phase is prophase, which you'll see pictured here. In prophase, there is a shortening and coiling of DNA into rod-shaped chromosomes that appear like X's because they are actually replicated chromosomes, also known as sister chromatids, and they are connected at their centromere. The centromere, also known as the centrosome, are the yellow things pictured in each diagram. Sister chromatids are identical to each other. There are 46 chromosomes at the beginning of prophase and 96 chromatids that exist. Centrosomes appear as spindle fibers. There are also hollow tubes of tubulin, and they are formed as the centrosomes begin to migrate to opposite poles of the cell. The nucleus and nuclear envelope are still visible, but it begins to break down during prophase. This is shown as the dotted line. The nucleolus is no longer visible. The next stage pictured is metaphase. In metaphase, the mitotic spindle has fully formed. Centrosomes, again the little yellow things, have moved to opposite sides of the cell with spindle fibers throughout. They're called centrioles in animal cells, but since we're talking about human cells, we refer to them as centrosomes. Then, replicated chromosomes have walked along spindle fibers. They are attached via the motor proteins in the centromere's kinetochore to the middle or metaphase plate, which will be labeled here, of the cell. They are in a nice straight line. That's how you know it's metaphase. Finally, the nucleus is no longer visible. The next phase of mitosis is anaphase. In anaphase, chromatids of each replicated chromosome separate at the centromere and move slowly, centromere first, towards the centrioles at opposite sides of the dividing cell. Picture the centrioles like a fishing pole, reeling in the chromatids, although it is the chromatids that are actually doing the movie, so don't be confused about that. Then the tubulin subunits break off as the section of the track is no longer needed, and they add to the non-kinetochore microtubules. This lengthens them, and this gives anaphase the overall look, which is why it's not the same circular shape that prophase and metaphase have, but rather a more oval look. The chromatids, once pulled apart from each other, are called chromosomes. This is important to note. This is now when they become chromosomes. They look a little bit sideways, and they all look like capital letter A's. We have now reached the last phase of mitosis. This phase is called telophase. In telophase, spindle fibers disassemble. Chromosomes relax into less tightly coiled chromatin shape. The nuclear envelope reforms around each set of chromosomes, and the nucleus forms in each set of newly formed nuclei. The little pinkish purple circles that are shown on this diagram are the nucleoid. One specific thing to note about telophase is that a cleavage furrow appears. This cleavage furrow signifies the splitting of the two cells, which will be seen in the next step, cytokinesis. Mitosis is now complete. Next, cytokinesis divides the cytoplasm by dividing the cell membrane. However, in animal cells, the cells divide by a cleavage furrow, and in plant cells, they divide by a cell plate that eventually becomes the plant cell wall. Cytoplasm and cell membranes are necessary for cytokinesis in both plant and animal cells. Now, in cancer, there are two genes to be aware of. The first is p53 proto-oncogene, and the second is the RAS proto-oncogene. However, when they're mutated, they just become the p53 oncogene and the RAS oncogene. When the RAS oncogene is mutated, it creates too much cyclin. Now that we know that cyclin helps carry on the process of mitosis, too much of it causes mitosis to move too quickly, and that's bad. When there's too much p53 oncogene, it is a tumor suppressor or regulator, which means that it would create, again, too much mitosis way too fast, which causes the duplication of cancer cells very quickly. You can see at the top is a normal graph of the way chromosomes go through mitosis. It's normal rates. However, you'll now see that when p53 oncogene goes through mitosis, it spikes way too high and way too fast, which means that it's going through mitosis much too quickly. You can see the same thing happen in the RAS oncogene. It spikes way too high and way too quickly. This means that it's irregular and too much mitosis is happening too quickly, which is causing the genes, the cancer genes, to duplicate way too fast. To add on to this, carcinogen is a substance that's capable of causing cancer, which means it affects the cells and leads to uncontrolled cell division, and is what causes the spread of cancer to grow so quickly.